Okay, today we are talking about PyBioNet Fit, which is a parameter fitting software package developed here at Northern Arizona University and in conjunction with Los Alamos National Labs. The purpose of this software is to address questions surrounding intracellular signal transduction pathways. Um, my research looks specifically at the MAPK pathway, but there are also models of the PI3K, JAK-STAT pathways, and many others. There is still active research going on these signal transduction pathways, so new models are being written daily. This software can also be used for what I put in parentheses here, pathways like COVID infections, by treating the path of a COVID infection from going to an asymptomatic or a symptomatic patient or a hospitalization or a non-hospitalization, just like a signal transduction pathway. But specifically, this software was created for cancer research. And so you can see our question here in the bottom. Uh, like I mentioned in my graduate presentation, the big thing here is these flexible portions of the BRAF kinase that allow it to transduce a signal downstream to MEK when there is no mitogen present upstream. So you can see the premise here of a type 2 inhibitor inhibiting one half of the dimer and a type 1.5 inhibitor inhibiting the other half of the dimer. And so our study looks to use both in conjunction and silence this oncogenic signaling downstream to MEK. The motivation for this mainly came from the previous attempts at rule-based modeling. They weren't originally called rule-based modeling because they didn't include rules based on rate constants. What they did include were just the molecules and the protein-protein interactions. They didn't include rate constants, and this violated the second law of thermodynamics. For any Kp rate constant, so the association constant is what we call it, where two molecules come together, there's an equal likelihood of a dissociation constant, or a K minus rate constant, that was not correctly interpreted in previous attempts at modeling these pathways. So moving into the actual software, you can pass in qualitative or quantitative data and receive any number of outputs. There are a ton of algorithms that we'll talk about in a few minutes that can parameterize different models depending on the data that you have or the data that you think you'll have. The main output is a fitting output. And so down here, you can see a best fit with the concentration of an inhibitor on the y-axis and time on the x-axis. So this is a really rough overview because the workflow is kind of different for every lab, depending on the different plugins you want to use, very similar to QIIME 2, where you kind of have an option of the analyses that you want to run. But it mainly starts here with this BNG model that relies on a program called RuleBender. RuleBender has a GUI, so you can actually make little contact diagrams and interaction maps of your pathways. But what you do with this model is you take it through a partial network expansion and you make your hybrid model. From there, you can move into NFSim, which is a particle-based network-free simulation, and create an output trajectory. This output trajectory will create that same fitting output and send it back to RuleBender if you want a graphical output, or you can just look at the .gdat files and kind of plot your concentrations of inhibitors and phosphorylated MEK concentrations from there. There's a lot of other things you can do. The main principle of PyBioNet Fit is taking this big biological issue and making it nothing more than a math problem. And then all the program is really solving is this massive ordinary differential equation. You can see down here that there are a lot of other different outputs that you can choose. So you can output it back into SBML, the systems biology markup language. You can put it into MATLAB or MCell. So really, you can do whatever you want here. You can even go into Atomizer with your SBML files and create different types of figures. The background of this software is that it's originally written in Perl. Some plugins and modules have been updated to Python 3, but the base configuration files are still in Perl. You're able to parameterize biological models using enhanced or conventional BioNetGen language or the systems biology markup language. PyBioNet-Fit is huge, and it has a large suite of meta-heuristic algorithms for parameter optimization. So the one that we'll be looking at in the demo today is the differential evolution algorithm, but there's also particle swarm optimization and scatter searching. The bulk of the progress that's been made in the last few years has really been in the Bayesian approaches and in the uncertainty quantification side of the programming. So there's a lot of different bootstrapping modules, and then there are multiple different Markov chain Monte Carlo algorithms. So there is the Metropolis Hastings algorithm, and then there is a custom and brand new adaptive Markov chain Monte Carlo algorithm used for HIV dynamics and that COVID-19 forecasting. The last thing that I'm going to touch on is the biological property specification language. And so this is actually how we pass qualitative data into the software for parameterization or model checking. So just as a review, the BNGL language, this is just a graphical showing on the left here, but the actual rules in the BNGL language are here on the right. So we have some different values that we pulled from the literature. And then we have these little chunks that aren't kind of well defined with maybe hashtags or something to comment something out. But you'll see that we say begin molecule types and end molecule types. So within there, we put our proteins, and in here it's RAF and then an inhibitor. And then we would end that chunk. Then we'd move into compartments and talk about where we're looking at this problem. So in this case, it's the cytoplasmic compartment. And then we would end that chunk. We'd go into seed species. Observables is really where we kind of get into the nitty gritty. This is our output. So for this specific model, we want to look at the concentrations of proteins, so R to R, or inhibited proteins, RI to R. And then down here, this RI species is a fully inhibited dimer. So we want the model to tell us what the concentration of those inhibited proteins are. Then we have more outputs down here, which is more along the lines of what I was just speaking about. But down here we have our functions. And so this is what we want it to actually calculate for us. And it can be really confusing because there's a lot of different math going on here, but we're essentially looking at steady state activity of our proteins in the absence of an inhibitor, and then the activity in the presence of an inhibitor. So how do you actually run one of these things? You pass the software two files. The first is your configuration file or .conf file. And so the way I always thought about this file is that it's very similar to a script for a Slurm job. This contains the directories where your outputs go, the model specifications, certain run parameters, the algorithm types. So for instance, we'll use that differential evolution algorithm, and then whether or not you want it to print to the command line and save the data of your best fit. The other file that you pass to the software is your .bngl file. We primarily write rules in the BioNet-Gen language and not so much the SBML language. So you can do either or, but you need a model that is written in one of these biological specification languages. And so this contains your actual model, your molecular observations, any parameters, and any actions or functions that you want the software to perform. So any of the math that you want it to do. The outputs that you get, the main one is a GDAT file. This is similar in my mind to a CSV or a .tsv file. And so this is going to contain your actual values. So these are the things that you can plot and create those pretty graphs that I showed in my graduate presentation. So usually these are your X and Y coordinates over time. The next one is your log files. And so this is mainly a traceback for the model that you ran. And it's very useful for debugging and seeing where a model went awry, or if you maybe messed up a rule in your original BNGL file. And the last set of files that you get is sorted parameter files. And these contain, I just put V1 free here, but it contains all your parameters at each iteration for each object in your model. So it's a little more useful for full traceback. So you can see what numbers were changing when, and if it was an iterative issue or if it was a rule issue. Okay, so how do we actually know that this software is showing us anything biologically relevant? The inhibitors and the concentrations of drugs and proteins that we get from our model fit are then checked in vitro. This is a colony formation assay using Meljuso cells, so melanoma cells. The predicted dose for synergy from this trial was 5 micromolars of vemurafenib and 3 micromolar sorafenib, so a type 1.5 and a type 2 drug. And as you can see here, there are no blue colonies, so there was no cell division. These cells died, given that they're all melanoma cancer cells, especially compared to our DMSO control over here on the right. In vivo, it's a lot tougher. You have to engineer the organisms to have the mutations that we're investigating, so specifically that BRAF V600E mutation. Once you make your transgenic organism, so we inject this MITFA promoter driving EGFP, MITFA promoter driving MITFA right here, you can have this fish that will express melanoma approximately 60% of the time within the first 6 months. That's when zebrafish reach their true adulthood, and their lifespan's around 1.5 to 2 years, depending on the strain. So after you have these transgenic fish created, you expose them to those drug combinations, which would be this 5 micromolar and 3 micromolar dose. And then you would do a Western blot. So you'd extract all the protein and you would probe for total ERK, so all of the final signal transducing protein, and phosphorylated ERK, or active ERK. Ignore the long exposure. This is just an artifact of Western blotting, but you can see in the short exposure panel that there are certain combinations where there is no phosphorylated ERK compared to total ERK, right? So that ERK was not turned on. This is how we check in vivo whether or not the drug combinations that the model suggested are actually synergistic, or if they were just an artifact of the model. But most of the trials that we have run after taking drug concentrations from our in silico studies have proven the same results in vitro and in vivo. Okay, with that being said, we can move into the demo.
Okay, so moving into the demo, let's take a look at a few of the file types that I was discussing in the inputs and outputs slides. The first is this bng2.pl file. And so this is the BioNet Gen software written in Perl. So here is really all that it is. It's not a huge file. Most of it is designed to take user arguments at the command line and then do something with them. So the bulk of this file and the kind of complexity of it comes in right here with the arguments and variables containing those arguments. I will go into the configuration files and the .bng files in just a second. The first thing, if you were trying to follow along, is to do pip install PyBionet fit. I already have it installed, so I'm not going to run it, but this would use the Python pip installer to install the PyBionet fit software into your machine. After that, you want to run the export BNG path, and we're going to set this equal to a file that we can get off of the Los Alamos National Labs GitHub repository. And it's this bng2.pl file. So this is the main configuration file, like the global configuration file. And so if you get rid of the file name and you run this bng path command, you are good to go. BioNet Gen is now in your path. So in order to run a model, all I need to do is go into my PyBionet fit directory here, look in examples, and look in the demo. And so in here, you will see that we have an output file because I've already run this before, and logs because I've run this before. But we're really looking for this demo_bng.conf file. This is our configuration file. The other one that will be specified in that file is this parabola.bngl file. So in order to do a run in PyBionet fit, you call the software PyBionet fit like that. And then I'm going to do -C for the configuration file. And I'm going to tell it that I want that file to be bng.conf, just like that. Oh, 1 second. Let me path into this folder. Examples, demo. Okay, so we can see that those files are in here now, and I can now correctly call the configuration file. And so now you are doing a run in PyBionet Fit. It's that simple. To look at what's going on behind the scenes while that runs, this is why I said that the .conf files are very similar to Slurm job files. You basically specify the output directory where you want your outputs to go. You have your model specification line of code. And so this is the BNGL file that I was talking about. Then you have the algorithm fit type and the objective function. So here we're using the differential evolution model, and we are also using the chi-squared objective function. Then you can set your parameters, and this totally depends on your run and what you're looking at. In my case, I can just set the V1 through V3 free values equal to 0 and 10. And then you have the differential evolution parameters because I'm using that differential evolution algorithm. And just for the sake of really saving some time, I'm going to set the population size to 4 and the maximum iterations at 30,000. If I were running this on the Monsoon cluster, the population size would be much larger and my max iterations would be closer to 100 or 200,000. The last two that we have here are printing your output to the command line. I'm not a huge fan of it. It just takes up a bunch of space. So I set verbosity equal to 0. And then I want to save my best fit data because I want to plot it or do something with it after the fact. And so I'm going to set this save best data variable equal to 1. Here it's asking me if I want to overwrite my previous run. That's fine. The only other thing to look at here is the parabola.bng file. So the actual rules that we're applying to this model run. So you see that we have begin model, begin parameters. We have original values, and then these V1 free values that we will specify in our .conf file, molecule type, seed species, observables, and then our functions. So here is where all the math is being done behind the scenes. Then you have begin actions. The really only important part here is this ODE. So we know we're doing differential evolution because we're having the program do an ordinary differential equation and solve that fit for us. So if you look back over here, someone, I think at Los Alamos, was using hello in their print statements to kind of fix an issue with cross compatibility. And when they uploaded the files back up to GitHub, they left the print statements in, which is kind of funny. But it says that fitting is complete. So now we can look at our .gdat file, which here we can ls and we can cd into output. And so we can look in here and see that we have outputs. One of the main components of that output is the gdat file. And so in that gdat file, we have 3 columns. We have the time, we have an x coordinate, and we have a y coordinate. So if we were to graph these time x and y coordinates, we would get a nice parabola, which was the whole point of the demo. So with that, I will take any questions that you all have.